Hello. Hello and good evening. I'm Astrid Sass. I'm the head of school of the School of Pharmacy and Pharmaceutical Sciences. And it is my great pleasure and honor to introduce to you today Professor Nicki Panoskaltis, Professor and Chair of Personalized Therapeutics. This is a joint position between the School of Medicine and the School of Pharmacy and Pharmaceutical Sciences here in Trinity. Nicki is also an adjunct principal investigator at NIBRT, and I warmly welcome everyone in the audience, the Provost, Dean of Research, colleagues, members of the public, and also Nicki's parents. Who are visiting on this very special occasion to celebrate with us Nicki's inaugural lecture here in Trinity. Nicki has asked me to make this introduction brief, and I will try to do so, honoring her wish. However, I do have to mention some of her outstanding achievements and will give a brief outline of her very successful career as a clinician and scientist. And this is how Nicki likes to be referred to: a clinician scientist. Nicki has a distinguished academic career. She obtained her medical degree from the University of Toronto in Canada, and further clinical training in internal medicine, hematology, and bone marrow transplantation at the University of Rochester in New York, where she also received the American Board certification. She obtained her PhD from Imperial College London in 2004 for her works on the role of the immune system in acute myeloid leukemia and how this can be translated into new treatment options for patients. She remained as faculty in London until her move back to the US to Emory University and the Georgia Institute of Technology. Nicki came to Trinity in 2022. I think she was a very serious and very high-performing student. She met Sakis during her undergraduate degree in Canada. Although she looks more Greek than him, she is a Canadian Greek speaking it fluently. In college, she helped Sakis to pass his organic chemistry exams. She was first in the class in organic chemistry, actually not only in her class, but scored highest in the history of the university. They have been together ever since. She and Sakis have a very close professional relationship that is clearly scientifically enriching and mutually supporting. They are far more than the sum of their parts, with immense energies and a shared focus that works really well without personal competition, and both very driven by making things better. If you're interested in more details about their journey as collaborators, check out their TEDx talk from 2018, available on YouTube. It is very entertaining. Nicki's early academic career shows a lot of achievement with prizes and scholarships. She has won awards for clinical excellence, research, innovation, entrepreneurship. She holds several patents and is a fellow of the Royal College of Physicians in the UK. It is common for colleagues and collaborators to say Nicki is extremely hardworking and highly accomplished. Everything she does is to the highest possible professional standard. Nicki describes herself as a clinician scientist, a rare breed. Her chair title very well reflects her research and academic interests and her fundamental driver: personalized therapeutics and improving individual patient outcomes. Above all, Nicki is committed to the patient. During her time in London, Nicki had the uniquely challenging experience of managing the aftermath of the TGN-1412 first-in-human clinical trial. She managed the aftermath at every level: clinical, scientific, logistical, regulatory, legal, with astonishing integrity and strength of character. TGN-1412 was an immunostimulatory antibody, an anti-CD28 superagonist, which was infused into 6 healthy human volunteers as part of a clinical trial. This was before immunotherapy became clinical standard of care. All 6 healthy volunteers experienced rapidly progressive life-threatening critical illness due to cytokine release syndrome, also known as a cytokine storm, which required clinical care with a specific skill set, and it was Nicki who offered her expertise acutely and long-term. All 6 patients survived. Today, Nicki's office at TTMI in St. James's Hospital shows many signs of gratitude from patients and their families. She's kind and gentle with patients and extremely serious about doing what she can for them. The prevailing theme of her work, aligned to the chair position, is the personalization of therapy, that the patients obtain best outcomes by tailoring the therapy to the specific disease state of the patient. Eventually, this led to the funding of IMPACT, which is short for Irish Medicines Centre for Personalized Cellular Therapeutics, which will develop and clinically implement advanced cellular immune therapeutics tailored for the individual patient. A fantastic achievement not only for Nicki and Sakis, but also for the school and for Trinity as a whole. Nicki, thank you for all that you've done as a clinician, as a scientist, and will do in the future. The floor is yours. Thank you.
And that was short. Thanks, Astrid. Thank you so much for coming today to this lecture. It's my honor to see so many people here, and my honor to give you some semblance of what I've done in my career to date. And I'm going to take you on a journey. As a hematologist, my area of interest is the blood, and in normal hematopoiesis, which is the formation of the blood in the blood organ, which is the bone marrow located in our long bones in adults, but also of cancers of the blood, in particular leukemia, which is a cancer of the bone marrow and the peripheral blood, and how we treat patients better and can treat patients better by listening to them more. I'm also going to take you on a journey from bench to bedside and back again. And this involves observations at the bedside, how as a clinician scientist, clinician scientists use those observations to take what we know back to the bench, discover other options for our patients, and bring it back to the bedside to try to get better outcomes for patients. That's what it's all about at the end of the day. I'm also going to try and convince you that personalized therapeutics starts with Star Trek. Now, Star Trek, I'm a Trekker, and Star Trek was a fantastic show. Gene Roddenberry is as a visionary, he's one of my heroes. And he really took what we actually take as commonplace today back in the 60s. And people are actually using these types of things now, which is really phenomenal when you think about it. The space-time continuum was really mentioned in these old Star Trek episodes. And I take that as the definition of personalization. It's all about space-time continuum. Probably not as physicists understand it, but it's all about the individual, how space-time interacts, and how that individual, that N of 1, interacts in their environment over time. And that when you visualize that patient in that N of 1, in that time slot, that is just one snapshot. You have to take the patient on their course of treatment throughout in that space-time continuum. It's also about being in the right place at the right time, to learn lessons and to take those lessons into actions for how things can be done better. I started my career in Canada, in London, and Toronto, and Ontario, went to the US in Rochester, as Astrid said, went to London, came back to the US after Brexit, went to Emory and Georgia Tech in Atlanta, and then came back to European shores in Ireland. And at each point, there was instances of aha moments that were integral in going forward with what needed to happen on a personalization level and how to treat our patients better. And hopefully you'll see that throughout the presentation. My early environment, this is me at the age of 6, by the way, my early environment, being the daughter of Greek immigrants, was formed by the myths and mythologies of ancient Greece. And I know that's corny, but actually, Odysseus was a great hero to look up to in certain ways. He was a bit of a philanderer at times and had poor navigational instincts. But overall, Odysseus was a good hero. In the Iliad, the wily Odysseus created the Trojan horse and was dependent on the arrogance and the complacency of the Trojans to get that horse in, get the Greeks into the Trojan city, and they won the war. That led to a huge failure for the Trojans. Arrogance and complacency led to that. In the Odyssey, he took a 10-year journey to get back home. Don't ask me why. It was just the Aegean that he needed to navigate. And it was the resilience and persistence that led to his success in getting back home to his wife Penelope and his son. To me, Odysseus is the ultimate nuclear cockroach. He could withstand anything. I also thank my parents, Christine and Nicolas, who, Greek immigrants as they were, they tried their best for their children to educate them and instilled in us very basic moral attitudes. Accountability for one's actions, really important, and doing the right thing is often the most difficult thing to do, but you need to do it. As I grew up, I had career choices. I loved mystery novels. I was a voracious reader, still am. And I wanted to learn and apply what I learned about the ancient Greek and Egyptian and Roman myths, and maybe I could become an archaeologist. But going into archaeology school and university in Canada wasn't very appealing to me. I couldn't really go to the ancient sites. And then I thought, well, okay, maybe I could be a dentist. But looking in people's mouths every day, thankfully Brian O'Connell's in here today, just didn't turn me on. I then came across a story about the history of interferon. This was in my teenage years, 14, 15. And I thought, well, that's interesting. Maybe I'll be a doctor. I'll shelve that for now. And then I thought, oh, I love Star Trek. What about an astronaut? That sounds cool. And, Bones and Captain Kirk, that's great. Male role models, by the way. And I was told by our vocational guidance counselor at school at the age of 11, you can't do this. You need to be a nurse, a dental hygienist, or a secretary. And I was put into a typing class, which now leads me to be able to type 60 words a minute and has done me good stead up until now, but that was not because I chose it. And the one thing people know about me is if you tell me not to do something, I'll do it. So with the words of Yoda in my mind in 1980, do or do not, there is no try, I wrote to NASA at the age of 11. And I was chuffed. About 3 to 6 months later, I got back this huge envelope with a bunch of photos of astronauts, mission reports, and a 3-page letter to me saying what I needed to do to get into astronaut school. I thought it was brilliant. 3 pages telling me about benefits of being an astronaut, what kind of health care I would get, etc., etc. They also had the mission report of Apollo 13. A successful failure, they called it, in true American terms, a successful failure. And it was interesting to read the report and that of the astronauts when they said that I transmitted to Houston that we had a problem, and they knew that they were in serious trouble when the 2 pressure oxygen gauges, one said 0 and the other one was going down during this mission. They knew they were in serious trouble at that point. And I knew at the age of 11 that there are such things as successful failures. So I went to university in my town of London, at Western Ontario, University of Western Ontario, where I was able to go and work in the lab of Nick Sinclair, who was an arthritis clinician scientist, who my sister Angela was working and doing her MSc and PhD in. And she had cheap labor, me, during my high school years and early university years, cleaning pipettes, packaging them, harvesting spleens from mice, and homogenizing them. I realized then, aha, I love research. Immunology is cool. I hate mice. I then met also Sakis, my future husband, my collaborator, and with whom we've shared this research journey together from early years of our university careers. My mother said, you're going to medical school. I said, no, I'm going into research. And after 2 years of university, she said, no, you're going to medical school. So I said, okay, I will apply, but I'll apply once. And if I get in, I'll go. So I went to medical school in Toronto. And in Toronto, they had a really interesting, now this is 30 years ago, an interesting model for looking at the medical psychosocial interaction of human health and disease. Now this is something that we're teaching now and is new, but then was really interesting in that they came to this as a model in U of T, and that they understood that biology isn't everything, and they taught to us the environment really matters for patient care as well as for disease and health. Of course, we all learn in medical school, prima non nocere, first do no harm, very important element to ensure that you instill in students, and that holding a patient's hand can be better than a bucket load of medicine. That was really an important lesson from Sir William Osler's work, a graduate of the University of Toronto, who eventually became the chief at Yale University thereafter. I also, as this was my personality then, wanted to go after the most difficult rotations just to make sure that medicine was really in my blood. After 2 years, I thought, oh, this really sucks. And then I started seeing patients, and I thought, oh, this is really lovely. I really like patients. I love patient interaction. And I did the most difficult surgical rotation possible, which was the transplant rotation at the Toronto Hospital, which was under the purview of the chair in transplantations. It's the big surgical chair in Canada, really powerful position. And at the end of my rotation, he calls me into his office and he says, Nicki, I hear from my team that you have a very surgical personality. And I said, I'm not sure if that's a compliment or an insult, and I really hate surgery because I find it really boring, so no, thank you. And that kind of burned bridges with my surgical career, but I had a calling to medicine, I knew that. So I continued my training at the University of Rochester in upstate New York with greats in the field, as you can see there, Bob Betts, Ray Dolan, Bill Hall, who really were fantastic in teaching the next generation. They were really committed to it, more than I've seen anywhere else, and that it's important to have the power of observation and critical analysis of data that's presented to you. 95% of diagnosis in any patient comes from the history, a good history and a good physical exam. Don't need any of these fancy tests. They're very helpful indeed, but the history and physical exam is paramount, and you need a good history. The patient will tell you everything. Really influential in my career was in 1996. This was at the end of my internal medicine training when there was a death of a nursing student in a clinical trial. It was a volunteer trial at the University of Rochester, and she was having a bronchoscopy. This is a procedure that goes into the lungs, just to look around the lungs, take up some fluid, and examine it. And she died due to an overdose of lidocaine. At that time, I think they were probably required to, but they did intensive training for all of the trainees at the University of Rochester to ensure that we knew how to protect our study subjects and how to take proper informed consent, how important it is, and what to do in the case of a serious adverse event. Who knew? A serious adverse event, what to do in that situation, medical legally, clinically, etc. They also instilled in us that the gold standard of patient care includes training the next generation of students and trainees. Truly important, truly valuable, and something that I tried to do throughout my career. Pay it forward, as you will. I also discovered I'm a lumper, not a splitter. And for anyone who knows what that is, it means you lump symptoms into a diagnosis instead of splitting them up into separate diagnoses. So I knew I was someone who was bringing things together. And I knew at that point that translational patient care, centered care, is in my future. I stayed at Rochester for my hematology and bone marrow transplant fellowships and did some lab research there with giants in hematology, Marshall Listerman, who was a past president of the American Society of Hematology and pushed us towards our bone marrow organoid model in the beginning days. John Bennett, who was a father of the acute leukemia and myeloid neoplasms classification system, is a pathologist, very well renowned. John DiPerzio, whose research group was instrumental and reported first on the clonal evolution, so all the mutations that happen in cancer, leukemia, and how that evolves over time and through treatment, and how it's different in every patient. It's personal to the patient and personal disease. Jacob Rowe, father of leukemia, et cetera, all of these greats I was training with. They taught me a sense of humor is really valuable, especially when you're dealing and treating patients who may not have long in this world. That involvement of patients in clinical research is a gold standard of care. I can remember rounding and being in clinics with these greats, and they would have clinical research studies to hand, and no patient was discussed without discussing clinical research, how to involve them in what was being done in research in the center, and how to offer them new medicines through clinical research, a really important pipeline. I sat in a clinic with Aaron Rappaport here, who was new faculty at the time. And I remember him sitting down with a patient quite clearly saying to him, okay, you have a new diagnosis of lymphoma. Your chances of remission are about 60% right now. No, sorry, I got that wrong. Apologies. Step back. Your chances of remission are 0% or 100%. And I said, but Dr. Rappaport, his chances are about 60%. He says, no. The chance of the cohort with lymphoma is 60%. But that patient, that individual, his chance is 0% or 100%. And that flagged, gosh, this is personal. This is personalization. This is about the individual, not about the cohort. And so it really frustrates me when I hear a lot of clinical colleagues who say to patients, oh, your chance of remission is 60%. No. Your chance of remission is 0% or 100%. And then we go to the next stage. The next stage after you've had your treatment is 0% or 100% again. That's the individual, the personalization. I also learned there that if you can't treat the condition, you've got to help the patient towards a good end. Hugely important for the patient and the family. We can't win them all, unfortunately. When I was an intern, and Jacob Rowe, who always rounded at 4 a.m. every day, he came in to do his rounds at 4 a.m. He was such a busy clinician scientist, came in at 4 a.m. every day. And he found me with his patient, 19-year-old with acute myeloid leukemia. Again, cancer, devastating cancer of the blood and the bone marrow. And she had relapsed many times, and she was on her last hours of life. She was bleeding. She was bleeding to death with her family at the bedside. It was devastating. I was doing everything possible as an intern. She was resistant to all the blood products that we gave her. She wasn't responding to anything. And the way that he dealt with the patient, holding her hand, holding the family, crying with them, has touched me to this day. And was the reason I actually went into hematology. I have to thank Dr. Rowe for that. He was incredibly touching and instilled into me the joys of hematology in that you help your patients through this, you help the families, you try to do your best with what you know, and you try to make things better for the next generation of patients. So I knew that AML, acute myeloid leukemia, would be my focus, my disease area of interest, and that there must be something better than chemotherapy, transplantation, and recurrent blood and platelet transfusions. This is my Star Trek brain talking now. What we're doing now with chemotherapy is barbaric. Surely there's something more elegant and targeted that we could give our patients that's less toxic for them and more effective. At that time as well, Sakis was doing his PhD in the lab, and we developed the first bone marrow organoid for leukemia during our training and patented it. This was almost 30 years ago. People are now trying to do this. Again, we've got the patent, it's there. I also did separate research in the hematology lab with Jane Leesville and Camilla Bood on trying to develop a cell therapy and a vaccine using dendritic cells. These are immune-stimulating agents, trying to create a response in patients who had leukemia so that the disease doesn't come back, so that they achieve remission. You can give them a vaccine just like we do for other infections and diseases, and that if you could give them a vaccine, this won't come back. Now, we weren't the first doing it. There were a lot of people who were interested in dendritic cell vaccines at that time. They're now interested in it again, so it comes in leaps and bounds every 30 years cycles of research. But I took that idea that I developed in that lab and went to the dendritic cell expert at Imperial College London, Stella Knight, at the Androgen Presentation Research Group. She leads that group, and she taught me, I have to thank her as my PhD supervisor and mentor, she taught me how to think as a scientist, how to write as a scientist, and that words matter. And I say words matter because for her, that was really central to her scientific career. She actually discovered the dendritic cell before Ralph Steinman, who got the Nobel Prize. But she and Bridget Balfour, who discovered it, called them veiled cells. They didn't call them dendritic cells. And so the dendritic cell won out, and Ralph Steinman got the Nobel. Words matter. And I learned during my PhD studies, I'm not going to go through any of that data, that leukemia, even though we thought all cancers, including leukemia, was a condition that did not have a stimulated immune response, I found that there were signals of inflammation in patient blood and bone marrow leukemia cells and their T cells, the cells that respond to immune stimuli in patients. Their immune system was activated. And I thought, well, that's odd. We think right now that the immune system is not activated. This is actually something that in the last 5 years, people are looking at more and more, that there's an inflammatory condition in leukemia that makes the leukemia evolve. So I thought, well, what am I doing making a dendritic cell vaccine? I can't vaccinate patients if I don't know what they're doing. They're already stimulated. How can I use a vaccine or a cell therapy unless I know what the immune responsiveness of that individual is? How do I know how they're going to respond? So I need to quantify that. I need to phenotype that. And it led also to the suggestion that if leukemia is an inflammatory condition, at least partly, can it be preventable? Can we dampen that immune response to see that the leukemia can actually be controlled, or in fact go backwards so that you don't get leukemia at the end of the day if you have certain signals that lead you towards that propensity? And that was a really heretical thought back then, 25 years, that now people in the last 5 years are saying, oh gosh, maybe it could be preventable. We needed, though, better models to look at this. We couldn't do this in the culture conditions that we developed already, the 2D flat culture conditions in flasks and plates. We couldn't do this in the animal models because the physiology of animals are very different. It's very artificial. So we needed a better model. And so Sakis and I, that at Imperial, got our heads together and said, right, we co-founded the Biosystems Engineering Lab, which is an interdisciplinary lab. And our feeling was that we could create a pipeline from lab to clinic with this interdisciplinarity between engineering, which is what Sakis is, and medicine, me. And integrating our efforts, we could then make an applied program for the benefit of patient applications. That was in 2003. Again, something that people are doing now. Back then, it was considered, again, heretical. And that integration could go towards personalization. So I'm going to talk to you today about what we did in the lab with our in vitro models, looking at functional precision models in our organoids with normal blood and leukemia. I'm also going to talk to you about the bedside, the patient observations that led to a lot of the research that we then evolved in the lab, mainly in the Tegenero trial, as I'm going to go through. And then the bench to bedside, which is integrating what we saw in the lab with leukemia growing in these organoids in bone marrow outside of the body. What we saw at the bedside with the kinetics of responses, how patients were responding to certain stimuli, and putting that together in a personalized immunochemotherapy program so that patients would have better outcomes and reduce toxicities. I'm going to start first with this bedside model and the Tegenero case, which Astrid has already introduced you to. So I think about TGN-1412 as the thalidomide of immunotherapy. Everyone probably knows about thalidomide. It was an awful drug when it was given many, many years ago, and it led to a lot of changes in regulatory oversight of drugs worldwide. And that's what TGN-1412 is. In 2006, in March, there were a lot of headlines in the media once this drug trial became known, the outcome. Drug trial creates elephant man, seriously ill men after UK drug trials fiasco, drug trial man's brain on fire, exclusive elephant man to lose all fingers and toes, tighter controls needed to prevent elephant man, drug trial horror. And this was the event in March 2006 where 6 young, healthy men almost died after they were given an antibody in a first-in-human study. These were healthy people, not patients. They all almost died almost simultaneously. This drug was given every 10 minutes on March 13th, starting at 8 a.m. And by midnight, they were in the intensive care unit. I won't go into the details of the drug itself, what happened, etc. It's not important for this talk. What is important is that at that moment, midnight 12:03, I got a call from Andrew, Costello-Cortez, here, intensive care unit extraordinaire, who's a bit of a joker, and he said, Nicki, I have 6 guys about to die on me. I need your help. They got an anti-CD28 superagonist antibody. I need your help now. And I said, now, at that time I said, Andrew, I've got an amateur inspection coming up. I'm the only consultant hematologist in the hospital. I'm on call. I've got all this stuff going on. You're on one. You're giving it to me. This is a joke. And I was about to put down the phone. And he says, no, Nicki, wake up. This is serious. We have a situation in our hands. I had just taken over as Director of Research and Development at the Trust some months back. I was the only consultant hematologist in the hospital. And this was the most humbling experience, I have to say, in my career. It was the point in time that many of us probably have in our career where all of the experience and expertise comes funneling down into this one moment. And I knew that I had to get from my training, my expertise, and my background what I knew about dendritic cells, T cells, CD28 is a molecule that this was stimulating, and everything I knew about patient care, clinical trials, and that clinical trial that went bad in Rochester to help me deal with this study. It also showed that what arrogance and complacency can do in terms of when things go bad, things go very bad. So TGM-1412 is an antibody to stimulate a protein on these immune cells. The details, again, don't matter for our purposes today. And it was supposed to stimulate a type of T cell called a Treg in the body. This is an immune dampening cell. So if you had an arthritis, for example, that was a stimulated immune response, a disease, this was supposed to stimulate the Treg cell in the body to dampen down that response. And preclinically, so in the lab, on the bench, in mice, in rabbits, in monkeys, in rats, this worked brilliantly. Arthritis was down by 80% in the cohort, again, and it worked brilliant. No side effects. Toxicity profile was well managed in all of these animal models, and they said, brilliant, this is all we need to go into the human. And that's what they did. They did a phase 1 single-center, double-blind, randomized, placebo-controlled, single-escalating dose first-in-man trial. Now, anyone who knows anything about clinical trials knows that this is ridiculous. You don't do a randomized, placebo-controlled trial in a first-in-human study. They were allowed to do it in the UK. The sponsor was Ted Gennaro, a company, a spin-out company from the University of Würzburg. The contract research organization that carried out the trial was Parexel. This was an American company. They rented space at Northwood Park Hospital where I was a consultant hematologist. And they got into trouble with these 6 people on the day of infusion. Now, the details of what happened after that is only important in terms of the outcome. All of these 6 patients had toxicity due to the stimulation that was unexpected from this drug called the cytokine release syndrome or cytokine storm. Completely unexpected because they were arrogant about their data preclinically. And we took that unfortunate incident, and this is the first report of cytokine storm explaining it from the beginning all the way to recovery 30 days later and beyond 2 years. And the first report of what happens in a cytokine storm to the cytokines, the cells, and the patients. All 6 patients thankfully survived. They all had prolonged recovery. And I think that if even one hadn't survived, this would have put immunotherapy back many, many years. Now remember, this is before any immunotherapy, this is 2006, before any immunotherapy was available to patients at all, okay? It also formed a reevaluation in the UK of phase 1 trials with new medicines, new biologics, new active agents. And at that time, the Secretary of State for Health, Patricia Hewitt, formed this expert scientific group, which was chaired by Professor Sir Gordon Duff, to formulate a report. Now, their edict was to not look at accountability. That was not within their remit. Their remit was to look at the future of medicines development in the UK so that it would not fall backwards, because this was the incident that would make it falter. Not only in the UK, but worldwide. At this point in 2006, where there were lots of immunotherapy trials ongoing, everything stopped, and the regulatory authorities had to stop everything on their desk pending the outcome of these 6 patients, pending the outcome of what happened in this study. Their remit was to cover the preclinical and early clinical developments so that things couldn't go wrong in future, the process of preparation and review of trial applications and advice of regulators and sponsors, determining and administering initial doses in man, so how much is the first dose and how does that relate to what you use preclinically? Is there an equal correlation? How do you look at that dosing schedule? The clinical environment for first-in-man studies and developing the skills and training to meet future needs. And I would argue that the UK, being a leader in advanced therapy medicinal products now, is because they put all of these things into place early on, right after the De Janeiro trial, and they were able to utilize these clinical environments and developing the skills and training to meet the future needs of new medicines. And that's why they're a leader in this field currently. Now, I remember standing up to the expert scientific group. I gave evidence many times. And at one point, they said, okay, well, we know that the ICU doctors did their ICU thing. They did their acute medicine thing. That was what really led to these patients surviving in the first couple of hours. But how did you, Nicki, how did you know what to do? And I thought, gosh, I don't know. I just knew. And that was just based on the experience from Rochester, good basic medical care, what to do when something goes wrong, really wrong. Also, my background in immunology didn't hurt. This fundamentally changed the conduct of first-in-human clinical trials around the world, this event, and the aftermath and the handling of it. And it enabled immunotherapy as the current gold standard of care, which millions of patients have now enjoyed the fruits of that, and some gone into complete remissions and cures, I'm sure. I always get very angry when I talk about TGM-1412 because we as a community, clinicians, scientists, regulators, etc., fail to protect the volunteers. The scientific rationale was flawed, the preclinical testing was flawed, the design of the trial was flawed, the regulatory safe checks didn't work, the conduct of the trial was flawed, the monitoring and follow-up of a serious adverse event was left up to local measures, and the legal and media handling was also left up to local measures. It was all local. De Janeiro missed the boat on immunotherapy. If they had done a good clinical trial, they could have been a contender and they could have been a blockbuster with this drug if it had been done correctly. It's part of that valley of death. They didn't do things correctly. They missed out and tried to speed up the process by speeding up the process. And it shows you what arrogance and complacency could do in leading to failures. They were arrogant and complacent about this drug based on the preclinical data. That resilience and persistence can pay off. It can lead to successes. And this really amounted to, I think, maybe a successful failure because things then improved for higher risk agents around the world and how we implement them and how we try them and make them safer for patients. So in addition to having this New England Journal report, and people say, oh yeah, but you got a New England Journal report out of it, aren't you lucky? It's like, no, actually I'm not, because this was 3 years of my life with 3 other busy full-time jobs, plus this, dealing with full-on 6 individuals and all of the regulatory, media, and legal issues. But it did force us. Jeff Drazen, who was the editor of the New England Journal at the time, said, we need this report as soon as possible. It's an ethical responsibility that we had to deliver it because it had to present the data objectively so that the clinicians and scientists dealing with other immunotherapeutics and the regulators could evaluate it and so that other therapeutic trials could go ahead. What really irks me is that they didn't look at the data. So they still thought that the T cells were stimulated in this, even in the editorial that goes along with this paper. But the T cells weren't stimulated. They got it all wrong. And further consequent reports and scientists who've done further analysis of what the superagonist does in human cells shows that actually the T cells were not the target in humans. It also led to a novel approach to the clinical pathologic immune correlates, a holistic approach to following patients in clinical trials when something goes wrong, to include things like evaluating for brain fog. Remember COVID? That's what these patients got. Cytokine release syndrome causes brain fog, just like COVID did. And long-term monitoring. Before this trial, it was only up to 30 days, and now it's even longer, because we know that there are long-term consequences. And those long-term consequences is seen in this busy slide, which shows you just what a cytokine storm here, you can see different kinetics for different proteins in the cytokine storm, leading to different kinetics in the cells that respond. And that's different for every patient. So it goes towards personalization of what happens in that patient over time. This is over a year, but really our analysis went over 2 years. Just to one stimulus. These are healthy people, not patients with a different disease. So you can imagine disease patients with different types of conditions will have different kinetic responses. It also led to thoughts of dynamic treatment options. If we could capture each of these responses, we can treat specifically and in a personalized fashion for a better outcome and to look at immunotherapeutic potential treatment options, in particular with chemoimmunotherapy for our patients with leukemia, which is where my mind was at the time. Other lessons, number one, when the excrement hits the fan, pen hits the paper. And I developed a whole 10-point SOP. S does not stand for standard. And this was a lesson from Rochester in that when something goes wrong, you write everything down. 3 years of my life, every day, was writing things down. Doesn't matter how tired I was, you write it down because you're bound to forget it when people ask you about it later, and it's done me in good stead. Be humble about the data. Don't lull yourself into a false sense of security, thinking that things will be fine when you go into the human studies. Prepare for the unexpected, very important. Think outside the box. Use common sense. Pick your battles. You can't win them all. Focus on the one important thing, and regardless of what else other noise is going on, for me the one important thing was the patient. It always came down to what was best for the patient, doing what the patient needed at that time. Doesn't matter what else was going on in the hospital or in my head. And I take this advert for this production that was done in 2017 on what happened in TGN 1412, because it focused on the patient. I participated in it because it focused on the patient. I've been really rather cognizant to divorce myself from this event as much as possible, but I've come full circle now in trying to embrace it and trying to make the best of what happened at that time. Make sure you like what you see in the mirror every morning. Make sure you've done the right thing. That can also be the most difficult path. Create opportunities out of challenges. Difficult, but always very rewarding. And I took that into the NHS. So where I was a consultant at the hospital at the time, NHS were always too busy, much like here in Ireland. Just too busy, no thanks, I don't want change, I don't want anything to improve, no wheels on my cart, please. And we instilled what I learned from Rochester in terms of research and training and embedding that into best gold standard patient care. And really using clinical governance as a means to get there. And I put here pictures of our fantastic nurses and Cara, who is a consultant colleague of mine, and we worked together to really make a safe environment to progress what we needed to do for patients, building new units, getting more capital resource, and help and availing of charities like the Richard Thomas Leukemia Fund, who are really important in getting what we needed for our patients and in the research in the lab. And yes, this is Stephen Borthwick, who was captain of the England team at the time. They raised a lot of money. He was patron of this charity, and they raised a lot of money for us. We also used what happened with Ted Gennaro and created an opportunity, applied for a competitive bid for a clinical trials unit at Northwick Park for our hematology patients. These are patients with cancers, very devastating cancers of the blood and bone marrow. And that also led to a clinical research network for the northwest of London. 1/3 of the London population is what it covered. And these were all competitive bids through the National Institute of Health Research in the UK. So these were the challenges at Northwick Park. We got very poor media because of the Ted Gennaro disaster, but because we handled it so well, we were competitive and turned it into a challenge to get better clinical trials, better access for our patients, and a safe environment for our patient at a time. It led to very high impact papers with phase 2 and phase 3 trials, another phase 1 trial for our patients, giving them access to drugs. And a lot of our patients, because of the care we gave, were some of the longest living patients on these trials. And certainly we were the highest recruiters on some of these trials internationally. At the time, I also developed, based on what we developed at Northwick Park, hemato-oncology guidelines. And these are the leukemia myeloid neoplasm guidelines, which are the basis for, I led this working group, and forms the basis of the guidelines for London right now in this disease area. I interestingly also found that it forms the basis of the national guidelines in Ireland. So I've actually contributed to your national guidelines for leukemia. I was chuffed to see. Through this process of dealing with things in the NHS and the academic centers and dealing with the Ted Gennaro, I found that common sense is indeed a very rare commodity. And this is something that Peter Medawar, who is a Nobel laureate in transplant immunology and was the mentor for my mentor, Stella Knight, taught her common sense is a very, very rare commodity. So I've talked about leukemia a lot. I'm going to talk a little bit about the leukemia journey now, because I think it's important we understand how complex these treatments are. And I'm going to take you through a hypothetical patient that I call Emily. Emily's a 45-year-old with 2 young children. She starts with her symptoms of fatigue, bleeding, and a fever. 2 weeks later, she decides finally, and after a vacation, she decides finally to go to the GP. She has some blood work, and the GP tells her, go immediately to the emergency room, you have leukemia. She gets a bone marrow. Her treatment is planned over a couple of days. She gets intensive chemotherapy over 10 days, and then the next 20 days, she sits in a hospital room waiting to recover because her immune system is 0. She needs supportive care with blood and transfusions, and she needs all of the support possible that's provided by the inpatient hospital environment in order to protect her during this time of intensive treatment. Unfortunately, she has resistant leukemia. She does not go into remission. About 85% of our patients will go into a remission, 15% will not. Of those who go into remission, most will relapse. There's a 30% chance of cure at 5 years in these patients. It's not a good condition to have. That's why there's better treatments that are needed. She has resistant leukemia. She gets more treatment. 30 days later, she relapses. She has a remission for a little bit after some more treatment. She comes back. She gets more chemo because she's relapsed. She has side effects with her relapses, more chemo. Eventually, she succumbs to her disease about 6 to 9 months later. This is not atypical. And so our bench to bedside approach in our interdisciplinary lab had to focus on how can we make our bedside solutions better? How can we make our supportive care solutions, transfusions, better for our patients? How can we make our treatments better, chemotherapy? There's got to be something better. Remember my Rochester days. And so our bench solution was, okay, well, my engineering colleagues say that we need to make blood in a blood factory. We need a bioreactor that's going to make blood. This is our engineering solution for you. The leukemia program was a little bit more subtle and said, okay, we'll use your little bioreactor, but we're going to make better treatments in vitro and in silico. And so we use this precision personalization based on the technology for looking at normal blood and leukemia in our program. So the blood factory, our own blood factory, we make hundreds of billions of cells every day, just sitting here. You're making hundreds of billions of white cells, platelets, and red cells, just to sit here and listen to this talk for, I hope, just under an hour. It's daily, it's dynamic, it comes in waves, increases when we need more, like when we're bleeding, and it's got a certain set perfusion rate. So we surmised that we could reverse engineer the bone marrow, provide a specific structure that was looking like the human bone marrow, enable the function by putting sticky proteins on it, and enabling supporting niches. So how do we make all these different blood cells grow together so that we can make a functional organ outside of the body, making blood? And we had 3 talented students, Mark, Teresa, and Susanna, all working at different times, who took the polyurethane, basic plastic model that we created over many years, perfected into looking like human bone marrow and putting the sticky proteins on, and we're able to use cord blood, a product that usually is thrown away at birth, doesn't harm the baby or the mother, and use that cord blood product to make red cells in the lab, and in a continuous process in those little static cubes by recharging the culture so that we could set up the environment in the first go over a couple of weeks and then put in another cord blood and make it run for as many weeks as we wanted. We had to terminate at 68 days because the students were getting a bit flustered and we wanted to finish their PhD. So, but we could make red cells that looked like human red cells in one-step process. And that was unheard of at that point in a heterogeneous culture without any protein additives, no cytokines and no serum. We put perfusion onto that and showed that human blood could be grown long-term in the lab, months, without the serum or cytokine, no additives. It was a very nice, non-artificial system, similar to human bone marrow, and led to this human bone marrow organoid for which we had put in a patent application. We used the personalized blood organoid to grow primary leukemia cells. We showed that we thought we'd get very similar kinetics, but actually every patient was different. Every patient we put into the organoid had different kinetics. We know that now. It's like, okay, they're all very heterogeneous, but then it was like, wow, how can we make one organoid to study all of leukemia? We're going to need different types of processes to look at each individual person in a personalized manner. We also found that the cells were resistant within the niches, these little niches here that were in the scaffolds. They were resistant to chemo and hypoxia, which is what happens in human leukemia. They hide in the bone marrow in these little niches. The chemo can't get to them, and that's what happens in the relapse, and that's what happens when the cells change. They've hidden in these little niches, they change in there, and they come back fast and furious, and you can't treat them. When we went to Emory and Georgia Tech in Atlanta, we met up with our colleagues, Doug and Deb, who were working on a PDX model of human leukemia, and that was a patient-derived organoid. So you take the human leukemia cells and you put them into a mouse that can accept them in order to keep growing the leukemia cells and test it for mechanisms of action, what makes the leukemia grow, and testing drugs. Now this is all great, it doesn't always work. It takes a long time to generate them, and you can see here it takes a lot of mice over many, many months to actually get one experiment through. We thought, okay, let's use our scaffolds. Let's see if we can propagate these PDX leukemia cells from the patient, these are patient-personalized cells, in our model. And we could indeed not only propagate them over months, but we could capture different phases of leukemia growth and different metabolic shifts, which were like the metabolic shifts that human leukemia cells go through when they have changes through relapse and through different treatment phases. It also made its own marrow microenvironment, which we also saw in the normal, and I haven't gone through. But this was the first PDX AML organoid model. We can replace mice, and we could use it in a way to show that there's better models of preclinical drug testing, which is what was required off of the Ted Gennaro case. We need better preclinical human models, not mouse models, human models, because that's what's going to make personalization, and that's what's going to make drugs better for testing in humans later. Okay, so now I'm going to go to the bench to the bedside. How do we get what we've learned at the bench with the leukemia models and the normal blood production, with what we've learned at the bedside and the kinetics, to the patient and personalizing treatment for leukemia? Excuse me. I'm going to take you back to Emily and her leukemia journey, which was not a good outcome. But our goal of personalization here is to get her at this point of diagnosis, get her a personalized treatment plan so that she doesn't go into those little niches and hibernate and come back fast and furious and relapse and we can't treat her for the life of us. And personalize that timeline so she gets a remission in a closer time point. We get all those nasty leukemia cells in the niche and we treat it all away. Now, this is beyond precision, so not targeted to the gene mutations, it's personalization. Yes, you're targeting abnormal cells, you're targeting mutations, but you're also personalizing it for the timeline of the patient and the patient needs. Now, at the time we discussed this, there was a lot of activity that we were doing with modeling of leukemia cells and the heterogeneity of cells, and my engineering colleagues in the lab were really getting quite arrogant, which I'm sure that you'll all understand. And they said, oh, we can model leukemia, we can model chemotherapy, of course we can get this done. You'll have a model in a month. Yeah, I don't think so. You don't understand. You don't understand how difficult leukemia is. You don't understand how heterogeneous leukemia is. We know that there's a call for personalized medicine from the NCI, the European Commission, and anyone else who's funding cancer drugs, and that it's going beyond the precision of mutations. And I was trying to explain to my engineering colleagues who thought they could model anything that we're dealing with heterogeneous patients with different body habitus, different sexes, all have leukemia, but they all have different mutations in their leukemia. It's all AML, as indicated here by the different colored dots. But we all give them the same chemotherapy, mainly, the same base core chemotherapy. They all get the same thing. They might get a non-intensive therapy, but that's a standard treatment because they're overweight or not clinically able to take the harsh intensive treatment. Based on that, they will go into either a remission, you can see here, where there's smaller numbers of cells, or they'll not respond to it at all, they're refractory. We then give them another round of treatment. The refractory person gets something different. These ones get the same thing again, different dose. And you realize at the end of this that that person at top might have been underdosed because he was overweight, and perhaps he would have done better with a different schedule, a different chemo, or a different dosing system. This patient in the middle was probably overdosed. He was lean, he got the right amount, but maybe he's going to have side effects and toxicity later because he got that amount. So he's going to succumb to something else in 20 years' time, like cardiac disease. And that third person who's refractory stays refractory. We still don't have any options for that person, and they're going to die. So personalized medicine really takes those three individuals, gives them a schedule of medicine and the type of drugs that they need according to their cells in the tumor and their own cells in the body, the normal human bone marrow, to optimize that outcome, a personalized treatment strategy for each of those patients, which is not something that is done now. And so my engineering colleague said, I see your point. Let's take you up on that challenge. And we had a very talented postdoc in the lab, Ana Quiroga from Chile, who looked at this and said, right, heterogeneity. We worked on a population balance model, and we could capture the heterogeneity of leukemia cells based on this mechanistic, it's not AI or machine learning, this is a mechanistic model, which worked on the cell cycle and looked at the clonal evolution and the different mutations in the different cells. This is now John DiPerzio's work from Rochester days. Different mutations as they evolve through treatment. The cells, the leukemia cells, evolve through treatment to change, and that's why it's so difficult to treat. You're not treating the same thing at every stage. We put that together with the PKPD module, so what the effects of each of these drugs have on the cell cycle in all the cells, different cell cycles at different times, different drugs. And we also captured what normal cells were doing in the bone marrow at the time, so that we knew that with different drugs, we would achieve a complete remission and that there would be recovery of cells at a certain time. So we could capture and simulate what the leukemia was doing and what the normal recovery was doing at that time with certain drugs. We also incorporated an immune module, to look for immune responses and incorporation of immunotherapies. This work led to a patent application and a spin-out company that we called Sanica, made up of the co-founders Sakis, Nicki, and Anna, Sanica, and a fast-track phase 1, phase 2 SBIR-STTR application to the NIH for $2.5 million, total of $3 million non-dilutive funding we had for the company, which we left behind when we left the US to come to Ireland. A risk we took, and we hope that will pay off because this was a very painful risk to lose all of that money when we left the US. Getting back to the PICIT advisory tool, we call this tool PICIT, just using the inputs of the patient, the tumor, so the patient inputs the tumor mutations, and what we knew about the drugs that we were going to use, we could put that into the clinician portal. This is an interface for the clinician to use to input data, and come up with simulations to 1, 2, 3, 4, as many treatments as we wanted. Came up with a simulation of what that patient was going to do to that treatment. Were they going to respond? How long were they going to respond? Were they going to recover? Did they have toxic effects, etc.? Non-intensive and intensive therapy, and could even see if the patient was going to be refractory, so we could exclude certain treatments from the beginning. We then had an optimizer tool, which looked at the clonal evolution. Remember, as patients go through treatment, they evolve their mutations, so they change the mutations, making it more difficult to treat when it comes back. Looking at the evolution and optimizing that for a better treatment in order to get into remission and hopefully cure. And here we have an optimized dosing for a patient who actually went through chemotherapy, showing that we could reduce certain chemotherapy agents, and by changing when they're given, not on the usual 10-day schedule, but giving a lesser dose and incremental in days 1 to 3, as we can see here, and then more of it about a week later, we could actually improve the outcome, reduce the leukemia cells to 0, and improve the toxic effects by ensuring that the good white cells that had to come back after 30 days could come back faster and stronger. So we could improve outcomes and reduce toxicity by using this mechanistic simulator and optimizer model. Part of our NIH funding was actually getting this into a clinical trial, a phase 1 trial. And this was based on the fact that the data that we collected was something that we could collect normally at the bedside. From the patient, we collected the blood. We also collected bone marrow, and these were standard tests that were being done that we inputted into the clinician portal, and the patient general assessment, body weight, etc. After it was put into the portal, the data would be processed behind the scenes, and there were treatment simulations and optimizations, which would then be on an interface with the patient. The clinician would go through this with the patient. The patient would say, this is what I want in my treatment. I don't want to be in hospital for 6 months of my life. Yeah, I understand what you're talking about for leukemia. I'm going to have better treatment options with this or with that, but this is what I want for my treatment journey. And this was something that could be put into this prospective clinical trial, and was put into it, as I said, funded to the tune of $2.4 million by the NIH, something that I was told here by my clinical colleagues wouldn't work. It was funded, the NIH thought differently. This led to an N of 1 clinical trial design and personalized precision treatment protocol that we can then go into novel clinical trial options and could form a step change in clinical practice. So how do we get back to the bench and the bedside with our new medicines and personal advanced cell therapies? Thinking about what we've done in the lab, what we've observed in the clinic, and what we know about personalization and in silico modeling. Well, the bench to the bedside is known for this valley of death. TED generics succumb to it. It's always difficult to get things from the preclinical phase into phase 1 and then beyond phase 2 clinical trials. 90% and over of drugs candidates fail. The cost is $2.5 billion, and the worst case scenario, like TED generic, you have a good drug, but you haven't done it well. You haven't tested it well. And the challenge and opportunity was to personalize, to create personalization in therapy. A lot of people ask me, what's personalized medicine? It's different for everybody. It's like the scientist with the elephant. You see a different thing, the blind scientist with the elephant. You see a different thing, you think it's something different. Really what personalized medicine is very simple. It's the right drug with the right patient, the right dose, the right time, and the right trial design, looking back to the space-time continuum. Now, Saki says that space-time continuum with me means that I don't have enough time to do what I'm doing, and I don't know what time is when I'm trying to be somewhere, like tonight when I was about to be late. But it's actually about the personalization, and it's about all of these things being in the right time, in the right place, getting the right drug and the right dose. So, since we arrived in Ireland, over the last 2.5 years actually, we've been putting together this personalized advanced cellular therapeutics bid for a center to Research Ireland. And really it's to enable patients in Ireland access to novel, effective, and affordable personalized advanced cell therapeutics. Something that we know Irish patients have difficulty accessing are new drugs. And this involves 16 co-applicants, 61 PIs, 7 universities, 5 hospitals, and 4 organizations in the Republic of Ireland, plus Northern Ireland as Impact NI. It's been my absolute privilege to work with all of these individuals so hard, long hours, night and day on this, because we really believe all of the individuals who are on this Impact Center application really believe in what is needed here in Ireland. What are cell therapies, one might ask, for those of you who are not in this field? It takes blood, there's that word again, blood, from a patient, puts it into the lab, engineers and modifies the cells into something that's targeted for a specific condition, like a cancer, and then takes those cells as a living drug and puts it back into the patient and it targets those cancer cells after it's infused. But the living drug is exactly that. It's a living drug. It changes. It changes its effects once it's in the human. And so patients can have very bad side effects if it's not treated properly in professional circumstances, like cytokine release syndrome, one of the major side effects of cell therapies. When we put this in, this is February, this is our fantastic research and development office staff, Kieran, Lynn, and Chris here, who helped us. This is a day of submission of this bid. We were a bit off our rocker at that point, but we got it in. And this is the team at the interview on September the 15th. Sakis is saying, please show us the money. I'm not sure why Walter was cut off the picture here, but I've included him in. And we were all very joyful going into the interview. Sakis, of course, is trying to strangle me on the way out, but that's okay. That's a different story. We made it through in the end. In that, although I can't say what the outcome of this is because it's on a communications embargo, I can say that I look forward to working with all of these individuals over the next 8 years. And it's not that I say it's important, or these 19 co-applicants say it's important, or all the 61 PIs on this bid say it's important to get cell therapies and personalized medicine into Ireland. Let me show you why it's important.
My name is Teresa Kenna. This is my daughter Erin, who's now 6, and my husband Joe. Erin was 4 weeks old and she was diagnosed with acute lymphoblastic leukemia, went through a lot of treatment. 4 months after Erin had her bone marrow transplant, we found out she relapsed. Thought we had no more options bar palliative care. Her consultant mentioned about this CAR-T cell therapy, had made a call to Great Ormond Street Hospital. When we went over to get her CAR-T cell therapy in August of 2019, the 5 of us travelled over together. We had to travel over and back for a year after for follow-up appointments. It worked amazingly well for Erin when we really thought there was no hope of anything working for her. The odds weren't in her favour. So CAR-T is amazing. I hope it becomes available for lots and lots of different types of cancer.
Advanced cell therapies will not just be limited to cancer, and the opportunity to apply them to immune-mediated diseases is something that we're highly interested in. Sanofi, with world-leading immunology that exists within Ireland, hope to bring new therapeutic options to patients.
Catalonia is leading the development of CAR-T therapies, and we are very happy to collaborate with the IMPACT project, foster an innovative and vibrant initiative to globally create a network where we can facilitate the access of the patients to this kind of therapies globally.
Advanced cell therapies are the future of medicines. I know that Ireland has the expertise required to innovate the next generation of these new medicines for patient benefit.
Ireland is ideally positioned to leverage its world-leading biopharmaceutical manufacturing expertise and talent for the manufacture of these new medicines. IMPACT will ensure that Ireland remains at the forefront of medicine manufacture, bringing further foreign direct investment and help build a homegrown industry.
It's so heartbreaking for us to know that there are Irish patients who could benefit from new medicines like CAR-T therapy, and they just don't have access to them. And it's why we support IMPACT, so more patients could have access to new, effective, affordable and personalised cell therapies. And so we ask, if not now, why?
Sakis is always happiest in the lab, and I need to log him in at some point just to stay there and do his work. To our teams, our research team in Imperial College at Georgia Tech and now at Trinity and Nivert. Many of them are here tonight. Thank you for coming. The patients in particular who have offered their samples for us to use and to learn from in the lab and for enhancing our clinical trials abilities, NHS staff, NHS Presentation Research Group at Imperial College, who I have to give them a shout out during the Tengenero event. They, without any funding, just came in and did all the immune monitoring, and Stella was brilliant with what was to hand. The IMPACT Consortium, as I said, a privilege to be working with all of them over the last 2 years, despite many arguments. That's also made us stronger. NIVERT, who've supported us with a lot of resource, and many, many others who are not listed here, collaborators, and in particular my parents, Christine and Tony, who have always been supportive and patient when I keep telling them I'm moving to a different country. Thanks.
That was absolutely amazing. Thank you so much, Nicki. I'd like to invite you to a reception now in a knowledge exchange area over there to the left. So thank you so much again. Thanks.